Hello there and welcome back. In this video we're going to be talking about how to solve or how to find gene distance in a single crossover event. Now let's say there were two genes, P and H. The parents, well, one of the parents were homozygous dominant for P and H, the other was homozygous recessive. Now when they were crossed, all of the F1 progeny were purple, meaning they were probably heterozygotes. Now let's say all of the heterozygotes in the F1 population, let's say they were crossed again with another situation where the parents would be all recessive, little p and little h. Well, in this particular situation, what kind of progeny would we be expecting? So mind you, the parents are going to have, one of the parents at least, the F1 generation, are all going to be heterozygous like this, big P, big H, little h. And we're crossing it with another situation. Let's say they were all recessive. So what would we be expecting if we crossed this? Well, an easy way to find out what we would be expecting is to use simple, smaller Punnett square. So let's just focus on one of the genes, and let's cross those together. So we have big P, little p is over here. And what kind of progeny would we get here? Crossing them around. These are little p's, mind you. And what you see here is that it's a 1-to-1 ratio. Now it's hard to properly determine such things because I have bad penmanship skills. However, these two here are homozygous recessive, meaning they're both two little p's. And these other ones are heterozygous, big P, little p. So what you see here is actually a 1-to-1 ratio. And that's what we would be expecting with the H if we cross these two. So overall, if we crossed a heterozygous F1 generation, big P, big H, little h, with this, something that's totally homozygous recessive, this particular situation also is called a test cross. So if we did this, we should expect a 1-to-1 ratio of F2, because mind you, this right here is the F1. So let's look at the F2 generation that we got. Notice that the numbers of each phenotype are very different. These two are the same, kind of similar. We can almost say these two, the 220 and the 210, are 1-to-1. But no, 220 to 32, that's not 1-to-1. They're varying very significantly. So why is this happening? Well, in this particular situation, this is the instance of a linked gene. These two genes, P and H, they're linked. And that means there's crossovers happening that are influencing the phenotype. So let me erase this and make myself some more room. Then we can talk about how we identify certain aspects like parentals and which of the phenotypes are actually crossovers. So in a population like this, where we're dealing with two genes being crossed, only two genes, nothing more than that, and you get a F2 generation that looks like this, the lowest amounts of the F2 generation, those are your recombinants. Recombinants it means the same thing as your crossover events. So these two phenotypes here, they're the result of crossovers. So if we wanted to get the total amount, well, actually, let me just write this down. These two here are crossovers. Specifically, they're single crossovers. And they're single because in this particular situation, it's more likely that you're going to get a single event of crossover. So what about these other ones? Well, notice that they're actually these are parentals. And they're parentals because they're recreating the phenotypes of the parents way up here. These are actually the same phenotypes, 220 and 210. The purple hairy and the red smooth, those are the phenotypes of the parents up here. So for the first question, it asks us to find the recombination frequency. So what is the recombination frequency of in this particular situation where we only have a single crossover event? To calculate this, we add up all of the crossover amounts that are occurring in here. Well, we know that these two are crossovers, so we add those up. 32 add 38. These are all of our crossover events. Then we divide it by the total amount of progeny, or the total amount of individuals in our F2 generation, which is down here, it's 500. So we divide it by that. And if you did the calculation, this adds up to 70, and you would get 0.14. Or you can say it's 14%. There's 14% of recombinants occurring in this situation. So this is actually the answer to the first question. So what about the gene distance? And when we say gene distance, we're talking about the distance between P and H on the chromosome. Because what you find with these linked genes is that they're normally found on the same chromosome. Genes that are on different chromosomes, they're probably not linked. So what is the gene distance between these two, assuming they're on the same chromosome? So if we draw pretend this line here is a chromosome, and we have gene P right here and gene H over here. What is the distance between these two? We don't determine distance per se, but what we find is relative distance. And by relative, I mean we know the distance between P and H on the chromosome, but we don't know where they are on the chromosome. And the distance is equivalent to their recombination frequency, which we got already. It's 14%. Now, we don't just write 14% as a distance. It actually goes in the units of either what they call centimorgans, or they use map units. So there's two kinds of situations or units you can use. So I like to use centimorgans. So we write, this is how you denote it. It's a little c and then a big M. This means 14 centimorgans. And you can also write it as 14 map units. I think it's MA or something like that. But I'm just going to use centimorgans. So now we can say that the genes P and H are 14 centimorgans apart. And another note that I want you to remember is that the order of the genes doesn't necessarily have to be big P or P then H. It could also just be H then P. We don't exactly know their order. And what you will find when you're dealing with 3 genes, not just 2 genes, when you're dealing with 3 genes, you can actually calculate or find out which gene is in the middle, but you can never really find out which one's on the ends. Thank you for watching. If you have any questions, feel free to ask in the comment section below, and of course, I do hope you're having a fantastic day.